So today we're going to be discussing the pathophysiology of Parkinson's disease. It is unknown the exact pathophysiology of PD, however it is believed to be resulting from the aggregation of the alpha-synuclein protein, which is the ASIN, the mitochondrial dysfunction and oxidative stress and neuroinflammation. So in order for this disease to occur, we have our risk factors and causes, which are non-modifiable are genetics, age and gender being a male and age 65 and over. Our modifiable are environmental and head injury. So from these risk factors and causes, we can firstly see that it can lead to dysfunctional protein clearance systems. So these are the ubiquitin proteasome system, which is the UPS, and the autophagy lysosome pathway. So these are the primary systems for the removal of dysfunctional proteins. The UPS is responsible for breaking down abnormal proteins. It does this by tagging them and transporting them for the degeneration in the proteasome. The autophagy lysosome pathway is divided into three. So first is the macroautophagy, where the autophasome engulf the intracellular components like the systolic proteins and fuse with the lysosome, which breaks down the content. The next phase is the microautophagy, where the cytoplasmic components are engulfed and destroyed by the lysosome alone. Finally is the chaperone-mediated autophagy, which is the CMA. This is a more selective process which involves the specific proteins being the target of the molecular chaperone, and they transport them to the lysosome for degeneration. ASIN is normally cleared by these primary systems. However, in the pathogenesis of PD, there is a damage to these mechanisms which cause the accumulation of the defective proteins of ASIN. Next, we can see the risk factors and causes cause oxidative stress and mitochondrial dysfunction. So oxidative stress leads to the impairment of cellular function. ASIN also interferes with this through its interaction with the mitochondrial membrane, allowing it to accumulate inside the organelle. Oxidative stress begins through reactive oxygen species, which is the ROS, production and detoxification, as well as the mitochondrial respiratory chain as the primary source of ROS. Therefore, due to its large role in ROS, the dysfunction of mitochondrial can lead to a decline in ROS, as well as the decreased energy production and the beginning of a stress-induced apoptosis. The gene mutations of SNCA and LRRK2 and the DNA of the mitochondrial can cause disruption in its fission and its fusion, leading to the destruction and deficiency of mitochondrial cells. When oxidized, mitochondria is taken up by dopamine neurons, and it leads to dopamine neuronal death in the SNPC due to energy depletion of the mitochondrial. As well as this, Parkin and PINK1 work together to facilitate the clearance of damaged mitochondrial. So when these genes have mutation, it causes dysfunctional mitochondrial to not be removed when they should be. Next, we have as our risk factors and cause is leading to neuroinflammation. Neuroinflammation is an inflammatory process that contributes to disease through the engagement of the immune system initiating cycle. This is through the microglial and the complement activation, T lymphocytes and pro-inflammatory cytokines in the SNPC, and the statium being of increased concentration. This mitochondrial activation can be a cause of dopamine cell death through the SNPC through pre and post neurotoxic insult. This suggests that the microglial induced inflammatory processes can be a cause for the lack of dopamine, as well as ASIN also being a trigger for this inflammatory process. So we can see that this neuroinflammation, which is also linked to the ASIN, is going to lead to neuronal loss. So we can see that everything kind of links to our ASIN protein and what actually is the accumulation of ASIN. So due to the imbalance of the synthesis and clearance, which we have here, SNCA gene duplication and mutations. We have a progressively accumulation of ASIN throughout the brain. It's found misfolded and in long filaments referred to as Lewy bodies. It begins in the dorsal motor nucleus, vagal nerves, and the anterior olfactory nucleus. These lesions progress to the brainstem until reaching the neurocortex. The combination of these mechanisms accelerate primarily dopaminergic neural death in Sabangia, Nigeria. This loss of dopamine production causes an imbalance through the basal ganglia between direct and indirect pathways, leading to the manifestation of the clinical symptoms, both motor and cognitive. And this is because this dopamine depletion, there is too much beta activity and too little gamma activity, creating different oscillatory rhythms. So that's what we see in our clinical manifestations, as you can see here, leading to Parkinson's disease. The most common medication used with Parkinson's disorder is levodopa, and it is a dopamine replacement, and its method of action is it's converted dopamine into peripheral tissues in the brain in order to replenish dopamine. And this is because in the pathogenesis there is dopamine neuronal death, and it's combined with benzoazide and carbidopa, which are both peripheral inhibitors, in order to minimize the peripheral dopamine production and reduce adverse effects. Its indications are Parkinson's disorder, and the nursing considerations are risk of aggravation of diseases. The following ones listed there: the history of substance abuse or depression, any treatment with dopamine antagonists, treatment with non-selective MAOIs, and giving it on time, because most symptoms persist quite quickly. Some side effects are nausea, hypotension, and vomiting, and this will be given to our case study just because she's experiencing a lot of motor symptoms, so it's likely that she needs a drug perfect like this. And these are the range of references I've used for both of these.